hospital where it's called twin star, and these proteins are very important for the development of any organism. We studied in the xenophys, the cell division. And then I want to talk a little bit about classical AD pathology and a form of pathology that Laurie discovered about 20 some odd years ago, which we call fulfillment pathology, and talk about its role in disease. And then I'm going fairly rapidly to cover some of these issues and want to talk a little bit about some of the spine labeling studies that we've done and the current applications of these that we're trying to take forward. So first of all, I want to tell you a little bit about the normal cellular functions of fulfillment, which essentially does everything. So the first activities of cofillin that were discovered were its abilities to sever filamentous actin and to generate new barbed ends of actin, so actin filaments get severed, and polymerization occurs from the severed ends, so you're generating nuclei for filament growth. But if the pool of actin subunits for growth is very small, then the filaments will depolymerize because you're also generating minus ends of filaments when they sever, and they can more rapidly depolymerize and come apart. Cofillin can compete with myosin 2. This is something that O'Neill has discovered in the lab, because cofillin binds to F-actin and can actually saturate F-actin filaments. And the saturated F-actin filaments are stable with cofillin bound. So how does it sever and how does it stabilize filament? It can sever, especially at low concentrations, but also in collaboration with other proteins that help in the severing activity. And this works especially well on long-actin filaments. Short-actin filaments, long-actin filaments. Short-actin filaments, if you have high cofillin, tend to get saturated and are quite stable pieces. These competition with myosin can change the nuclear architecture because actin filaments with myosin attached interact with the nuclear envelope, and I'll show you some dynamics going on there. Cofillin binds to G-actin and can be involved in nuclear transport. Cofillin gets phosphorylated and dephosphorylated. This is what we consider the active form, but that's only active with regard to actin. Cofillin in the phospho form, it's quote active inactive form, is an activator of the phospholipase D1, and this changes lipid dynamics in the membrane, and this can also play a big role in signal transduction. So it can feed forward into a lot of signal transduction pathways. Cofillin binds and also seems to have an interaction with ERP1, which is a protein that's involved in mitochondrial fission. So abnormalities in cofillin can lead to mitochondrial fragmentation, other mitochondrial dysfunctions, including mitochondrial-dependent apoptosis. So the oxidation of cofillin that can send it to the mitochondria can actually lead to cell death. And its oxidation can also lead it to being sequestered into cofillin-actin rods, which is this pathology that we talk about. But it also may be beneficial to cells to have this pathology because then the cell won't die because the cofillin is actually sequestered and not targeted to the mitochondria where it can lead to cell death. So it's a very confusing mixture of lots of different activities, and sorting them out is not a simple fact. So just to show you a couple of the things that I do, one is in directed growth cone motility. Cofillin is a major player in the actin dynamics of the growth cone. This is a pipette. Ordinarily, this might be a chemoattractant causing it to turn, but in this case, this is actually releasing from the surface of the pipette active cofillin that's encaged in a cell-penetrating peptide that allows the cofillin gradient to actually go across the membrane and cause this. So we know that it's active cofillin that's doing this because we can use the pseudo-phosphorylated form that's inactive, cage that, and we get no effect on the turning of the growth cone. So this is a direct turning response to the cofillin gradient that's actually penetrating the cell. You can see that cofillin, which is shown here with fluorescent tag, is at the leading edge of a migrating cell. and where it actually plays a major role in the system. In this particular cell, this is a image of the cell labeled with LifeAct. So this is the F-actin at the cell extending lamellipodium, and we've expressed in this cell, Eric vitriol, expressed the killer red version of cofilin. Killer red is a red fluorescent protein that has an alteration in the barrel structure so that water can get in, you generate reactive oxygen, and it kills. That reactive oxygen will bind to and kill anything that is attached to it, an enzyme that's attached to it, in this case cofilin. And so cofilin gets inactivated, and when you do that, this is a chymograph of this kind of line scan before photo activation, what we call chromophore assisted laser inactivation, Cali or microcali, and this is the time when the laser was activated and we killed the cofilin, and you can see the actin band spread, so it's not being depolarized. So these are some of the normal functions. What O'Neill has shown is that if you actually knock down cofilin in cells, you get massive blubbing due to hypercontractility. So this is where cofilin is competing with myosin binding, and as soon as you, so this is in the silenced cofilin. When you add blebbistatin, which is an inhibitor of myosin, it completely silences all of this blubbing activity in the cell membrane, which is due to hypercontractility. And it's really impressive when you see this in a dividing cell, which actually can't divide because cofilin is necessary for removing the actin ring structure that is involved in the cytokinetic furrow. And if you don't depolymerize this actin, it actually can't bring the membrane fusion of the daughter cells. So this is the chromatin in the nucleus. Look at the kind of stress that it's under being bounced back and forth between these daughter cells. And this is hypercontractility due to the lack of cofilin expression. Okay, so what are some of the other properties of cofilin and where is it important? It's very important in spines, both for their development and for modifications in spines that occur during long-term depression and long-term potentiation, which are electrophysiological correlates of various processes in memory and learning. The phosphocofilin cycle within the spine is regulated by a kinase that inactivates cofilin for its active dynamics and a phosphatase that will activate cofilin, and it can change cofilin dynamics. You can get rod formation under some conditions and it will inhibit myosin activity. But all these things can lead to change in spine structure and dynamics. And so we know that to go from a resting state, if this is a resting state of the spine, to actually get long-term depression by low-frequency stimulation, you actually get a hyperactivation of cofilin and the depolymerization of a lot of the actin that's in the spine, and you get spine shrinkage. And usually this is accompanied by some internalization of the receptors on the surface of the spine. If you do high-frequency stimulation or chemical LTP stimulation, you get a rapid activation of cofilin, which actually then leads to more nuclei for assembly in the spine of actin, and then you get a rapid inactivation of cofilin, and the phosphocofilin actually can be seen collecting in the neck of the spine, and you get a very stabilized actin filament structure that supports then a large spine area and makes more synaptic contacts with the presynaptic terminus, and this gives you what's called long-term potentiation, enhanced stimulation by electrical release. Okay, so mammalian neurons have a broad signaling pathways. So we're going to talk first about AD pathology. I think that slide was out of place. So classical Alzheimer's disease pathology is the formation of amyloid plaques and neurofibrillary tangles. And these are what pathologists look for in Alzheimer's disease. The A beta pathology in these plaques is formed from a peptide amyloid beta that's cleaved, self-assemble into these amyloid fibrils that have a nice structure that you may not be able to see there, but then they deposit in these insoluble plaques. Plaques really don't correlate very well with any kind of dementia. You can be cognitively normal and still have a lot of plaque burden in the brain. It may mean that you're headed into problems, but there is no correlation between the amount of plaque and the severity of Alzheimer's disease either. On the other hand, there is a fairly good correlation between a soluble pool of amyloid beta that's in the dimer trimer form and cognitive deficits. Neurofibrillary tangles are also looked at by pathologists because these things are so easy to see. So plaques and tangles, you can do a stain, and anybody can recognize these things in a slice. You don't need to be expert to actually see these things. And this is a picture that Lori took of immunofluorescent staining. There are these big flame-like structures of immunofluorescent staining. There are these big flame-like structures of tau deposits. Tau is a microtubule binding protein, a stabilizing protein, but one that seems to get released from microtubules during the process of development of Alzheimer's disease. What no one else looks at is cofilin pathology. Well, there are a few people now that are looking at it. So cofilin pathology, if we look at age-matched controls that we stain for cofilin in human brain, we actually see microglia that stain fairly well for cofilin, but you don't really see any what we would call cofilin pathology of really aggregated in blobs of cofilin, which we do see in Alzheimer's disease brain. And often these show up in the form of these rod-like aggregates. This is a section from human AD brain. Some of these aggregates get very large, that they're rod-like in shape, but other ones are definitely rod-like. And if we actually look at these in conjunction with tau staining, so tau is shown here in red, or phospho-tau, these are in what are called striated neuropil threads. And these neuropil thread structures are the same thing that you find in the neurofibrillary tangles, except these are out in the neurite processes. The neurofibrillary tangles come off of the cell body. And so these are a lot less obvious than neurofibrillary tangles, so pathologists usually don't quantify neuropil thread pathology. But if you actually are comparing the intensities of the green that you see in this AD patient with the intensities of the phospho-tau, you can see that in this case, probably cofilin pathology is fairly major. And it is, if you look at a certain region of the brain, and these are all done in the parietal cortex by Lori, who showed that if you look at non-cognitively impaired individuals, mild cognitively impaired, early AD, and AD patients, there is a progression. These are each individual subjects, and these are 10 fields of each quantified for rod pathology. You can see that most of these are above the controls, but obviously there are some in this region, at least, that didn't show any cofilin pathology, but there are also some in this region that didn't show any tau pathology. But these were classified as AD patients. So there are other regions of the brain that are obviously equally important as the ones we're looking at. But you can see that in some cases, in AD brain, cofilin pathology is equal or greater in terms of area than what you see in tau pathology, and yet it's something that no one else ever looks at. And in neuronal culture systems, rods will develop out in the neurites. They form in both axons and dendrites, and these rods can be seen in electron micrographs as bundles of, in this case, wavy filaments. These are glutaraldehyde before looking at it in the EM. Here's a rod in a neurite that is fasciculated with another normal neurite. So here's a microtubule in this neurite, and here's a bundle of these actin cofilin filaments. There's a microtubule here that kind of disappears when it gets up to where the rod is located. Lori has isolated these rods from neurons. It's a somewhat painful task because you have to manually lyse them all, and therefore you have to pass them through different size syringe needles, starting with scraping the neurons that have these rods off the dish. You have to depolarize the microtubules so that you don't end up with fragments of microtubule that are exactly the same density as rods when you do the sedimentation and isolation on gradients. But she's isolated these rods. They contain actin and cofillin, and these are cofillin dimers because of cross-linking of cofillin by oxidative stress seems to be essential in rod formation. So reactive oxygen production will induce rods. And we don't know the exact pathway of rods. You could get cofillin oxidized, forming dimers, and then binding to actin and coming together. My preference is that you probably get activated cofillin saturating actin filaments, so you get these stable pieces. And then under reactive oxygen, these come together and bundle into these processes. And that's because when we looked at these in vivo in EM, they consist of a wide variety of lengths of actin filaments, much like you would get if you just So Tom Kuhn gave you an introduction to this slide in his talk, talking a little bit about the role of TNF-alpha receptors and prion proteins and signaling for activating a transmembrane NADPH oxidase, giving rise to reactive oxygen species, which can lead then to localized cofillin oxidation and bundling of these filaments into rod-like structures. And this kind of signaling complex works with the HIV GP120 protein that he told you about. Amyloid beta, dimer trimers especially work through this pathway. TNF-alpha stimulates things through this pathway. And it doesn't seem like you actually need the TNF-alpha receptor for this to work. But are we 100% sure of that? Maybe not, maybe only 99%. But what we do know is that if you just overexpress prion protein, you generate spontaneous rods in neurons. So just increasing the amount of prion in the membrane seems to increase the sites where you get this reactive oxygen production, and then you get a lot more rod formation. The interesting thing is that you can form rods completely in the absence of the prion protein by using other inducers. So rods are also induced by glutamate. So excitotoxic responses don't require the same pathway. They're not prion dependent. So there are multiple pathways for rod formation and probably different pools of reactive oxygen. These pathways probably require NADPH oxidase. Glutamate excitotoxicity may require mitochondrial release of reactive oxygen. So different sources of this can lead then to rod formation. And you notice in this case, this is nearly 100% of the neurons that form rods. In this case, we can maximally, with overexpression of prion protein, get up to maybe 50% of the neurons with rods. And with normal stimulation with things like A beta, we usually don't get more than about 20 or 25% of the neurons. So there are differences. So in terms of Alzheimer's disease, are these rods important? Well, in a mouse model of Alzheimer's disease, David Kang's lab has shown that indeed you get rod formation if you're looking at the hippocampus or the cortex in the control, and in the Alzheimer mouse, there's a lot more of these red blobs which represent rods, and we quantified them in here. There's about a 10-fold increase in this rod pathology in these animals. And these animals can be measured for their contextual fear conditioning and their ability to remember a training exercise. And if you actually reduce rod formation, and there are two different ways that he did it. One is reducing one of the upstream regulators of the phosphatase that activates cofillin, and the other one is just reducing the quantity of cofillin that's expressed in these animals. They are normalized in their contextual fear conditioning, so their behavior is reversed back to normal. They don't forget anymore. You see actually a normalized LTP. This is the mouse brains and slices of the mice that actually electrophysiologically they don't really have LTP, but if you do them in the cofilin, the homozygous cofilin mouse or in the RAM-BP9 mouse, it's restored to normal LTP levels. So it does seem like cofilin rods have some major role, well, it seems like cofilin at least has some major role. The question is it rod pathology or is it just some other aspect of cofilin of the hundreds of different things that it actually does in the cell that are leading to some of the problems that may occur in cognition during Alzheimer's and I might add many other diseases of cognition. So what we wanted to do was to move from a dissociated neuronal culture system, which is interesting for studying cell biological problems, but doesn't really give us a lot of other interesting information. For instance, what's the role of glial cells that surround the neurons and how might they modify the response that we see? And being able to actually look at circuitry that exists rather than artificial circuitry that we develop through cell interactions in culture. The one reason why we wanted to go away from animals, number one, if we do all the animals we need for all these experiments, it gets very expensive. We don't really know the composition of the cerebral spinal fluid that we're bathing the brain in. And if we have mutant mice that are so-called A-beta expressing mice and develop cognitive symptoms, we have to wait a long time. We don't know the actual amounts that are present there. It's a lot easier if we can take these into culture and be able then to change the medium that they're bathed in, but still maintain the morphology that one gets in slices. And the other thing is if you're testing therapeutics and you really want to see can you reverse slices, you do need to go into brain, but you'd like to know that these things actually work in a system that isn't going to cost you an arm and a leg to just look at pharmacokinetics and transfer across the blood-brain barrier. So for screening things, having cultures where you can get like 20 slices out of one mouse and then being able to determine those response curves and then decide whether it's worthwhile going back to the animals, that makes a lot more sense. So we wanted to do brain slices for this kind of imaging. We also wanted to know whether or not we can observe pathological changes. So we've made a reporter construct for cofilin actin rods that doesn't induce rods. It's a mutated form of cofilin that we can overexpress in cells. It does not induce rods when it's overexpressed, but will label rods. It has an MRFP link to it. We also have one with an IRFP for fluorescent. And the other thing is that if we're lucky and we can do this on slices in culture over a long time period where we can observe them, we might be able to actually remove the slices from, our culture system and be able to do EFIDs. And we're hoping Sohan can work miracles on this and analysis. Okay, so what are the main methods of doing this kind of culture? A long time ago, two different methods were developed. First was this membrane slice culture where a permeable membrane was set up over medium. The slice was put on here, air is on the other side, and the slice was both oxygenated and fed at the same time. You could cut slices of about 300 microns of hippocampus or other brain regions and keep them alive for a long period of time. The other method that was set up was putting slices onto cover glass, cover slips, and inserting them into a flat-sided tube and rotating these so that the medium, fact that it's hard to do any kind of microscopy on the slices that are inside a tube like this. But this is a closed system, so if you're using viruses to express things in neurons, this is a great system to use. This is a lousy system because to observe the slices, if you're infecting them, you either look at them live coming down with a water immersion objective from on top where you can look down in the slice, or you have to remove the slice and put it on something else to look at and then put it back into culture. Contamination problems, microscope objective contamination, all these other things are possibilities. This one, if you fix the slice and pull them out, you can use them and look at fixed slices, but it's not a live system. So Each one of these has its pros and cons. The roller tube system, you don't really want very thick slices in there because if they're attached to a cover slip, you don't get oxygen penetration very well from both sides. But it's great for viral and for holding the slice before it adheres. And one of the things that you need to do is remove the plasma clot to get good imaging because it can lead to kind of cloudy, fuzzy imaging if you don't do that. But we have other ways of getting around that now. The nice thing about having the slices mounted on a cover slip is that you can use fiduciary marks to actually find the exact same cells that you were looking at over time. And I'll show you how we've applied that. The bad thing about these cultures when people first set them up is they'd use a good adhesive substratum and the cells would migrate out and the slices would thin and you'd lose all your morphology. So the campus would thin down to a couple cell layers and it wasn't very good. The air-liquid interface, there's no slice thinning, but imaging is more challenging and some of these things are fairly obvious. So what about slices? So we make the slices, we put them on cover slips, and the attachment process was iffy. First time that we did it, we coat the cover slips with a 3-aminopropyl triethoxysilane by dipping the cover slips in this, and it puts in a layer of silane on the surface that have amino groups sticking up that's bound into the glass. And from those amino groups, you can then try to make better adhesion with other kinds of molecules. But you don't want really good adhesion because if you put in things that promote cell migration, the cover slips, the slices are thin. So you want something that adheres the slice but doesn't promote migration. And one of the things that we've been using is this molecule called Vitrigel. It's one of these hydrogels that you can get either with or without an RGD sequence in the peptide sequence for it. That RGD are 3 amino acids that actually promote binding to integrins, and this actually promotes cell migration. So you find that in things like laminin and fibronectin and things that people used to coat the cover slips with that led to thinning. But you can buy this Vitrigel and it still polymerizes, but it doesn't have the RGD domain, and therefore you can get attachment, but you don't get migration. So that seems to be working well enough. We then this is the slice in here. We then attach the cover slip to the secure seal rings on the bottom of the drilled out tube, and now we actually have a way of looking up at the slice, live slices. They're not cultured under anywhere near this amount of medium. We use a few hundred microliters, maybe 600 or 800 microliters to just cover the slice. When these are tilted in an incubator, in a roller tube apparatus, they're at about 5 degrees, and the culture medium is in the back of the tube. As these tubes covers the slice and then uncovers the slice as it goes up and around. About every 10 minutes they rotate, and they're well aerated, and the slices stay alive. To view them, we use a microscope modification, a stage modification that Jeff told you about as one of his projects. He built us this tube holder for holding these tubes flat so that we can observe the slice through this cover slip. And we have looked at these on photo etch cover slips. So here's a hippocampal slice on a photo etch cover slip. This is grid number 34. The bottom of that 4 is right here. You can see it here. And so when you find that, you can do your XY coordinates to the region that you want to look at in the slice. And it doesn't matter how many times you take it out and put it back in. When you find the bottom of that 4, if you just go and push your buttons to get that same XY coordinate, you go back and you see the exact same population of neurons at day 14, day 15, 16, and 17. And you can actually, these are new O stained, which is a viability stain for neurons. You can find the exact same neurons that you want to look at. We've had problems with modifying this, but now I think we're lucky and we're hoping that these are worked out. Isaac will tell people a little bit about the method today when he does the workshop. So one of the problems that we also had was with our culture medium, and Lori spent roughly a year and a half or 2 years of her life trying to determine what the heck is wrong with these cultures. And it turns out, and the company that makes neurovasal medium, which is what we use, swore up and down that there's never been a change in the components of the medium. And that's probably true, but the source of all the reagents in the medium does change. And she came across some literature from a lab in Israel or Germany, that stated that they found a lot of D-serine in L-serine batches that were used for making medium. And you could not find a medium that didn't have low D-serine. D-serine is toxic to neurons. So Lori ordered all the components, made up every component from scratch, weighing out everything and making our own homemade neurovasal, and used a high purity L-serine that has less than 10% of the contamination of D-serine that is in normal medium. And voila, that seemed to help slice survival dramatically. She also discovered that L-cysteine in the medium is likely to be the component that's responsible for a problem we were having just in normal cell culture with high spontaneous rod formation. And so she's played around with changing L-cysteine anywhere from 10 micromolar up to about 260 micromolar, and has found optimal concentrations that really work for giving good viability, low spontaneous rods, but still allow rods to be induced by the things that we use, like A-beta. Oxygenation in these slices is important. We had a whole bunch going for Marcia's research here, and we had a power outage at CSU, what was it, July 5th. We had a power outage at CSU, what was it, July 5th. And of course, our incubator stopped turning for 2.5 hours. So on the 3rd of July, about 95% of these slices look beautiful. On the 9th of July, which was a couple days after the power outage, we lost most of the slices. And of these that were remaining, most of them died shortly thereafter. I think we still have a couple that are still alive. We have 5 that are still alive out of like 150 that were started when we started here. And so power outages can be bad. One of the other things, as you may already imagine, is that we want to be able to look at things in these slices like dendritic spines. We know how to look at spines. I mean, these are neurons that are growing on a bed of glial cells, but only the neuron is expressing a fluorescent protein here. This happens to be CEP-GluR1. This is a subunit of the AMPA receptors, and this supereclectic fluoride is the pH-sensitive GFP. So you only see it on the outside of the cell when it gets inserted, and if you drop the pH, the fluorescence pretty much disappears. What you're seeing inside is very faint pH because of what Phil said about the internalization compartments all being at pH 5 or at least less than 6. And so we can actually see new spines that are formed over here. This work was done in James Zane's lab. But Isaac has come up with a nice way of labeling neurites and being able to see spines very nicely with dialistic labeling. This is a dye called dye-I, which is a membrane label. A crystal of this and implant it into a slice where he wants the neurons to get labeled. He can follow the flow, the flow of the dye from that crystal out in the neurites. And we do this in live slices. So he labels it, we puts it in culture, and then we can keep them. This is 4 days, 7 days, and 12 days, going back to the exact same population of neurites using our photo etch cover slips. And the difference between being able to do this in a plasma clot and being able to do this in Vitrogel is quite dramatic. But in Vitrogel, we can actually get, with the spinning disk microscope, we can get very, very nice image stacks where you can see spines. We can rotate these images. We should be able to get measurements of spine density, spine shape changes data using the system where we can quantify spines. We can look at responses to A beta over time at very low levels. We're using about 1/1000th the level of A beta that most people use because they use synthetic A beta. We use A beta that's a secreted form from cell culture that we can fractionate, and the dimer-trimer fraction is really where the potency is for rod formation. So we're focusing on that. So I think I have one more or two more slides just to show you the fact that we've now been able to move from using postnatal pups for getting our slices to adult slices. So Lori has now been working on culturing slices from adult mice. I think these were like over 6 months of age. They're at post-breeding age of mice. And these are the new O is a vital stain for neurons, and this is an adult hippocampal slice. One of the nice things about the adult hippocampus is that we seem to be able to maintain much more of the entorhinal cortex and the subiculum going into the hippocampus proper, and this probably allows us to see much more regions, and with the adult slices, maybe be able to see age-related changes that are important in taking place. And I think I have a couple more slides, but I am going to stop right now and take questions. But I'm happy to entertain the questions.
So Jim, I think depending on the stimuli that leads to rod formation in hippocampal neurons, the percentage of cells that forms the rods are different, right? For example, A-beta 20%, glutamate 100%. That depends on the expression of the receptor on the cell, or which results in different levels of phosphorylation of coffulin or rod formation.
I personally don't think coffulin phosphorylation per se plays a big role, just because we found in adult brain that looking at the brain slice, somewhere between 3 and 7% of the coffulin is actually phosphorylated. So you're not going to get a huge increase in rods just due to a few percent change in coffulin phosphorylation. But I think a lot of the coffulin is kept inactive in another way, and that is binding to TIP2 at the membrane. Okay. And so I think you have a pool of dephosphorylated, potentially active ADF, but it's kept inactive by binding to lipid. And that could get released. And that release need at sites where we get reactive oxygen production, and that can then lead to its cross-linking and rod formation. All of the factors that work through the prion-dependent pathway, the GP120, TNF-alpha, A-beta, all of these things only give rods in about 20% of the neurons. And if we add them together, 20% of the neurons. So it's the same neuronal population. We're not seeing different neurons. That's why I think it's the prion patching that is involved with all of the signaling that's going on there. And that's why if you knock out prion proteins, none of them give rods. So it's all through a prion-dependent mechanism. The glutamate is completely separate. That is the same thing that I think we stimulate when we do in hypoxia and stroke. It basically leads to an ATP depletion, lots of reactive oxygen, probably mitochondrial source, and that causes rods in virtually every cell type.